See, this was what we had done in the last class. Everyone remembers this? Okay. Just wait 1 minute. Wait. Okay, let's start. So see, whatever results we have obtained in the last here in the last topic. Okay. So what all things we had seen here? First of all, what things are important? The formation of the gametes. You should first of all you should take in mind that whenever the parents or the zygote is formed, that zygote is a diploid cell. Okay, that means it has two n number of the chromosomes. Even number of the chromosomes are there. When the gametes are formed, so the chromosome number reduces to half. And if there are two types of, suppose if you have two types of chromosomes, so there will be two types of gamete. Okay, each gamete will carry only one chromosome, and the chances of the chromosome to be each gamete also here each gamete has an equal chance of fertilization. Now here it's not that the suppose here if you have a dominant gamete and a gamete containing a dominant allele and a gamete containing a recessive allele, so it does not mean that the recessive allele will not opt for fertilization. Both of them, they have equal chances of fertilization, like here we discussed. You see, in the case of male, you have two types of gametes. One is of the type capital T and the other one is of the type small t. So both of them, they have equal chances of fertilization, right? You see here, this capital T also has the same amount of zygotes. It can form two types of zygote, and this small t, it can also form two types of zygote. Understood this much? So this we will just study that what do we call this property. Okay, then the last one is the law of the dominance. Law of dominance means if the alleles, if the factors or the genes for both the contrasting alleles are present, so only the dominant allele is represented. The recessive allele is not represented out of it. For example, in this situation here, both the chromosomes or both the allele, both the chromosomes, they represent the dominant allele, that is tallness. Hence, you will get a tall plant. Here, one of the chromosome is for the dominant one and one is for the recessive one. Okay. So hence, in this case, you will get a tall plant. Okay, the dominant one will be represented, the recessive one will not be represented. You will not have anything like okay, you will not have anything like here that you will have a decrease of height or increase of height, or there will be a medium heighted plant. It's not that like that. A medium height plant will not be obtained. Okay, and The recessive allele, it will represent itself only when the dominant alleles are not present. This is the law of the dominance. See, Mendel's law of dominant, it states that when parents with pure contrasting traits are crossed together, only one form of trait appears in the next generation. The hybrid offsprings will exhibit only the dominant trait in the phenotype. Okay. So that means the first generation that we obtained, capital, which was had the genetic makeup as capital T and small t, that was a hybrid offspring, and it exhibited the characteristic only of only of the dominant trait in the phenotype, not of the recessive trait. Okay. So I think everyone has understood the law of dominance.
Yes.
Yes, sir.
Now see, here we did the crossing over at the last stage, something like this. The same thing can be done using Punnett squares. We can form here Punnett squares, and those Punnett squares, using those Punnett squares also, we can show the formation of the zygotes. So we form a square, divide it into 4, which is something like this. On one of the sides, suppose on the top we represent the male gametes, and here we represent the female gametes. There are two types of male gamete, capital T and small t. There are two types of female gametes, which are again capital T and small t. By the fusion of the gametes, here in the first one, you will have the formation of capital T and capital T. Here you have the formation of capital T, small t, capital T, small t, and small t and small t. Okay. So the same thing done using the Punnett square. From here, you obtain your ratios. You obtain the genotype ratio. We see here there are three genotypes. One is your capital T, capital T. The other one is capital T, small t. And the other one is small t, small t. Okay. So what would be the genotypic ratio?
1 is
1
to
is to 2
1.
is to 1.
Yeah, the genotype ratio is 1 is to 2 is to 1. You have 1, 2, and 1. This is the genotypic ratio. And phenotype ratio?
3 is to 1.
3 is to
3
1.
is to 1. Okay. Phenotype ratio was tall ratio dwarf, and it was 3 ratio 1. So here we studied the variation of just one characteristic, that is the height of the plant. And such a cross which we have studied is called as a monohybrid cross. Okay. So this cross we call as the monohybrid cross, where we have studied only a single characteristic. Okay. A cross involving two characteristics is called a dihybrid cross. So now today we will be studying about the dihybrid cross. Okay. So a dihybrid cross, it was made between two characteristics. So here we took pea seeds, and as we know that the pea seeds, they have a variety of variations in them. Okay. In the last class, I showed you that what variations can be there. So can you give me some of the variations what which we can talk of the seed shape and color, I mean.
Colors.
Right? So we discuss about here two things, seed shape and seed color. So there are two shapes which are possible. Either the seeds can be round shaped, or they can be wrinkled. In the color, there were two colors. Either you have the green color and you also have the yellow color. So these are the two things. We are going to study two characteristics together. If two characteristics are studied together, we call it as a dihybrid process. Okay. So what seeds were taken as the parent generation? Yeah. What seeds were taken as the parent generation?
Round yellow and wrinkled green.
Round yellow or round and green? Okay. What was taken?
Round green, sir.
Which one?
Round green.
Round green. Was it? So the first set was round and yellow,
And
and the
then
second
wrinkled
was
and green.
wrinkled and green. So these were the two sets. You took round and yellow. This was one set. You have wrinkled and green. So these were the two types of seeds which were taken, and they were made assured that they are pure ones. So you have pure round and yellow, and you have pure wrinkled and green. So these two seeds were taken, and out of these, round and yellow was taken out to be the dominant. So this was the dominant trait. Okay. In the
Sir, I have
shape,
a doubt.
yes.
Sir, why do we consider yellow color as a dominant color for pea seeds? Sir? Because like mostly we see the pea seeds are green in color, like almost everywhere.
Mhm. Okay. See what you see as your pea seeds, which are green color. See, your first question, why we consider them to be the dominant? Okay. We consider them dominant as the result of the first generation. When we obtain the first offspring generation over here, when we will be obtaining the first offspring generation, so we obtained only round and yellow. So this was
Yes,
from
sir.
the experiment. From the experiment, we obtained the first generation to be round and yellow. Okay. So from this only, it was clear that round and yellow is the dominant. Okay.
Right, sir. Yes.
Okay. Now your second question is you have always seen pea which is green and wrinkled. You have seen green pea leaves, green peas and not these one, not yellow ones.
Yes, sir.
Okay. So have you ever had those pea pulses? Pulses made of pea.
Yes, sir.
What is the color they have?
That is yellow, sir.
That is yellow. Because what pea you are eating, that is a green one. Okay, that is green. And basically, we try to eat those which are not ripened. Okay, once they ripen up, so the green ones will remain green always, and the ones which are yellow, so they will turn out to be yellow.
So what we usually have, most of them are not ripe, sir.
Yes,
So what we usually have peas, sir, they are not
yeah,
ripe.
yeah, they are not ripe.
Okay, sir.
So your both the answers are clear.
Yes, yes.
Okay. So see here to represent, as we know that the dominant characteristics, they are represented with the capital letters. So we start with our first P1 generation. Let us say in the P1 generation, again we have the male and here we have the female. And in the parent generation of the male, in the male parent generation, what we took here round and yellow. And female characteristics, they were taken wrinkled and green. So round shape, it was represented by capital R. The yellow characteristic was represented by capital Y. And hence the genetic makeup of the parent, male parent, was capital R, capital Y, and capital Y, as they always occur in pair. Manan,
Yes, sir.
are you getting?
Yes, sir.
Okay. Okay, so what symbol we will write for wrinkled and green, Manan? Manan, what
We
symbol?
will
Yeah,
small r and small y.
yeah. Here we will write here small r, small y, small y. Right? This is what we will be writing for this. Now, okay, Tanishka will tell. Tanishka, can you tell what type of gametes will be formed from the parent? From the male parent, what gametes will be formed?
So I think from round and yellow it will be R and Y, and from wrinkled and green it will be small r and small y.
Okay. So is there any other possibility of formation of any other gamete? Are there any other chances of formation of any other gamete?
No, sir.
Okay. Can't we have here something like this, only capital R, capital R?
Sir, but then the gametes, don't they have to be don't
They have
both
to
of
show a
the
color
characters
also.
have to be included?
Okay. Shresi, which law of Mendel gives us an idea that this is not possible? Only this is possible. Which law of Mendel gives you an idea?
Mendel's law of segregation.
Yeah, Mendel's law of segregation. This is Mendel's law of segregation. So I'll give you a statement for this Mendel's law of segregation. Okay. So just I'm writing here, this is because of law of segregation. This is due to the Mendel's law of segregation that you obtain two independent, two different ones. That is capital R and capital Y is possible, but this capital R and capital R is not possible. Okay. Then you will have the formation of zygote here. Formation of zygote will take place, and the new of generation we call it as F1 generation or the first offspring generation. So, Manan, what would be the genetic makeup of this first offspring generation?
It should be. Sir, I don't know.
Hmm.
I don't know, sir.
Okay, Vedika. Vedika, what genetic makeup will be there for the F1 generation?
Sir, capital R
Hmm.
and small y.
Capital R and small y. Is this
Sir,
okay?
capital
Is this okay or is there anything left?
Sir, it might be small r and capital Y.
Just let Vedika answer. Vedika?
yes, sir. And sir, capital R, small r, and
Hmm.
sir, capital Y, small y.
Okay. See both of them. You combine all of them. This one and this one both will remain there. So you will have your new generation, which will be capital R, small r, capital Y, and small y. So is this a pure or impure? Is this pure or hybrid?
It's impure. Sir.
It's impure, or we call it as a hybrid.
Hybrid.
It has a hybrid makeup. Now, since it has a hybrid makeup, so what will be its phenotype? Its genotype is hybrid. What phenotype will it have? Mehak, what will be the phenotype of this F1 generation?
So the ratio?
No, for this F1 generation, what characteristic do we expect from this?
Round yellow.
Round and yellow. Okay, very good. So this one F1 generation, we expect this to be round and yellow. So you do not have any mixing up of the characteristics, right? And Dhruv,
Yes, sir.
which law of Mendel now tells you that this will be round and yellow?
There are law of dominance.
Yeah, law of dominance. The law of dominance tells you that this generation or F1 generation will be round and yellow. Now, this is your F1 generation. From this F1 generation only, we obtain the first parent generation. This is your parent generation, second parent generation, or P2. We will also form the gametes of this. So this is the genetic makeup. With this genetic makeup, we start with the next one, that is P2. Again, you will have your male. You will have male plant and female plant. The male plant will have a genetic makeup capital R, capital Y, small y. The female will also have the similar makeup, capital R, and capital Y, small y. Okay. So, Saryu? Saryu?
Yes, sir.
Okay. Tell me the gametes formed. What will be the genetic makeup of the gametes?
Sir, it will be RY, capital R and capital Y.
For this one, capital R and capital Y. Is there any other possibility?
Sir, it can also be small r and small y.
It can be small r and small y. Yes. Any other possibility?
Sir, small r capital Y and capital R small y.
Okay, it can be small r capital Y, or it can be capital R small y, right?
Yes, sir.
Is there any other possibility?
Sir, I don't think so.
Okay. So, by which law of Mendel are we obtaining this?
Independent assortment.
Yeah, law of independent assortment and law of segregation. Both are here. We see here both of them. We see law of segregation as well as the law of independent assortment. Okay. See, why I say the law of independent assortment? Law of independent assortment means whether the dominant allele is present or not, whether the recessive allele is present or not, it does not make any difference. The gamete formation will take place equally. It will have the equal outcomes of the formation of the equal number of gametes will be. Law of segregation. That means whether there are single characteristics or there are multiple characteristics, okay, the characteristics, each characteristic is dominant by a single gene. One gene is dominating a single characteristic. Okay, so these are the gametes which will be found for the male. And Hasnita, can you tell what are the gametes that are formed by the female?
Sir, they will be the same as the male gametes, right?
Yeah, they will
Capital
be
R,
the same.
capital Y, small r, small y, small r, capital Y, and capital R, small y.
Okay, so here also you will have the same thing. Same as the male gametes. Now, our next step is the fertilization process and formation of the zygote. So by the fertilization process and the formation of the zygote, now you have 4 options in the male and 4 options in the female. Okay. So I would like all of you to make this Punnett square. Okay, where there will be four male gametes and there will be four female gametes. So I think everyone can make. K. Subha Charvik. K. Subha Charvik.
Yes, sir.
Can you make the Punnett square for them?
Yes, sir.
Okay. So I want all of you to make the Punnett square. All of you should form the Punnett square. Let's make the Punnett square over here. I'm also making, and all of you are supposed to make the Punnett squares. So everyone is making.
Yes, sir.
Okay. So just in 1 minute, I will be checking all your work. Within 2 minutes, I will see how much of the work you have done. Everyone, please keep your cameras on. So first of all, here you write. Generally, we write first the dominant ones. Capital R, capital Y. Then we write capital R, small r, capital Y. And at the end, we write the recessive ones. You can, if you write this one as first, so it will not be wrong, but just to have a good look and a better understanding, we do like this. You will have capital R, capital Y, capital R, small r, capital Y, and here you have small r and small y. Okay, to complete the squares, Dhruv, turn on your camera. Okay, sir, you can you show how much of work you have done? Okay, almost done, almost. Fine. And from that, take out the ratio. Find out the ratio. Capital R, capital Y.
Sir,
Yes.
how do you get the genotypic ratio for this, sir?
Genotype ratio, you collect all the genotypes. Collect all the similar genotypes. So generally the genotypic
For
ratio,
genotypes, sir, we have to see which is pure, which is unpure, and which is like that, right, sir?
see, there will be one set will be pure. Only one will be pure and one will be impure. So impure will be all. There will be two pure. One will be purely round and yellow, and one will be purely wrinkled and green.
The rest of them are
So,
impure, sir.
can I show
Yes.
you the? I'm done. Okay.
Yeah, I'll show it.
So can I show next, sir?
Okay, are you showing?
Yes, sir.
Wait. Let me unshare. Okay, this is Hasnita showing. Okay. So you have got the genotypic ratio. What is the genotypic ratio you have got? One ratio?
1:2:2:2:2:4:1:2 1:2:1:2
Again, show the ratio. Show the ratio of what all?
Sir, between the twos, I forgot to put the ratios, and that's why it's a bit messy. Otherwise, it's right.
Okay. See, if you are putting out the genotype ratio and even the phenotype ratio, put what qualities are there. For example, in the phenotype ratio, it's first one is 9 is for round and yellow. Okay,
Yes.
then 3
Okay,
is
sir.
for okay. So in that way, put those things also.
Yes.
And who else has done? Manan, have you done?
So yes.
Okay,
I'm
show
doing,
Manan.
sir.
Manan, you are still doing?
Yes, sir.
Okay. So this one. Yeah. Yes, Shreyasi, you also write the things. Write the characteristics also in the phenotype. If you have obtained the phenotype ratio, write the characteristics. The ratio is round and yellow to wrinkled and yellow. Then you have round and green, and the last one is wrinkled and green.
Oh, okay, sir.
Okay, then through. Okay, just I'm checking for Dhruv. Vedika, just wait for 1 minute.
Yes.
Okay, Dhruv, fine. Dhruv, write down the ratio. Okay, very good. Write down the ratio. And Vedika, now you can show. Yeah, Vedika is correct. Tanishka. Srilasya, you have not shown. Turn on your camera and show it. Yeah, just show it. Tanishka, show it.
Yes, sir.
Yeah, you also have not written that. And one more thing, Tanishka, just keep them a proper sequence. Like you have whatever gametes you have written on the top. On the top, if you have written the gametes, so the recessive gametes you write them at the end. Small r, small y, write them at the end.
Okay,
Are you
sure.
getting?
Yes, sir, I got it. So the dominant ones at the start and then the recessive one towards the end.
Yeah.
Okay, sir, I'll do that.
So can I show?
Yes, Mahek. Okay, Mahak, it's correct now.
Sir, I'm not exactly able to understand the
Okay, so
xenotypic ratio, sir.
okay, what you are not able to get?
The
Making of this,
xenotypic
making of the crosses or finding out the ratio.
ratio, sir. Crosses I've completed.
Okay.
And I even
So-
got the phenotypic ratio, but I'm unable to understand the phenotypic ratio part.
okay, okay. Srilasa, you can turn off. So it's not necessary to find out the genotypic ratio for the dihybrid one. Okay. Otherwise,
Sir,
you okay. I'm telling you how you can find out, but it will take time for you to find out each and every.
okay, sir.
Okay. So
Sir, in that case, can I just show only the phenotypic ratio and the...
yeah. Show it. Show the phenotypic ratio.
Yes, sir. Um, um, yes. At first, I tried doing the cross
No,
method, but
in that way you cannot do. That will be very complicated. We are going to have
I
16
started.
combinations. Okay. So 9:3:3:1. What is it? What does 9 represent? Write that also. What does
Okay,
this
sir.
3 represent? Write that.
Okay, sir.
Okay, good. Good. So with this, we have completed this portion of Mendel's experiment of monohybrid crosses and dihybrid crosses. So everyone has understood this? Yeah, I think everyone has got this how to make their crosses. So you need to learn the statements from here. You need to learn the law of dominance, law of segregation, and law of independent assortment. Right? So can anyone tell what does the law of independent assortment mean?
Sir, is it whether the dominant allele is present or not?
Okay. The alleles of two different genes get sorted into gametes independently of one another. Okay, so that means whether the allele is present or not, whether the dominant one is present or not, it will not make any difference. There will always be a independently. They will arrange them independent of one another. So this is your law of independent assortment.
Sir, can you repeat, sir?
Yes,
Sir, can you again explain it, sir?
yes, I'm giving you the statement. Just wait. I haven't shared the screen.
No,
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 sir.
no.
Okay, just wait. You see, the statement of the law of independent assortment. It states that the alleles of two or more different genes get sorted into gametes independently of one another. Okay. So you can say that the allele of a gamete receives for one gene does not influence the allele received for another gene. Means if suppose a gamete is there which is receiving a gene for the a color that's red, so it will not affect that whether it is also getting a gene for shape or not. Got this law of independent assortment? it basically applies to your dihybrid cross. Okay. It's not applying for the monohybrid crosses. As over there, it's only one characteristics. Here we have two characteristics. Okay, so this was your law of independent assortment. Can anyone give me the statement for law of segregation?
Sir, I have it, sir.
Okay.
During the formation of zygote, each gene separates from each other so that each gamete carries only one allele for each gene.
Okay, so during the gamete formation, what happens?
Zygote formation, each gene separates from each other such that each gamete carries only one allele for the each gene.
Okay. So during the gamete formation, each gamete receives only one allele. It cannot represent two. It cannot have two alleles. So that is your law of segregation. I will add it to your notes. Now with this, we are clear with what all Mendel did. Let's see the statements over here of if anything is left out from the book. Let's check. Okay, we are left with that sex ratio, right?
Sex determination, yes.
Yeah, sex determination we are left with. We are left with sex determination, and I think we are also left with blood group. These are the two things which are left from this topic. So let's complete today's sex determination. Sex determination, it tells us that how the sex of a newborn baby is determined. See, basically what I am writing here, instead of that, you also need to listen carefully each point of the sex determination. So, Vedika, how many chromosomes are there in human male?
Sir, 42.
42.
46.
46. There are 46 chromosomes. So
23
you have
pairs.
23 pairs. You have 23 pairs. 46, or we say it to be 23 pair. So out of those 23 pair of chromosomes, we represent them as 2n. So how many are the sex chromosomes? Out of the 23 pair of chromosomes, how many chromosomes are the sex chromosomes?
One.
One. There is just one pair of chromosome which is a sex chromosome which determines the sex of an individual. Rest 22 chromosomes, they determine other characteristics. Right? So out of the 23 pair, we are not writing all the 23 pair over here. We are just writing only one pair of chromosome which determines the sex of an individual. Okay. And these are two types. Either in the male, you have a chromosome of the type XY, and in the female, you have a chromosome of the pair of the type X and X. Okay. So these are the ones which control the genetic characteristics, which control the characteristics of male and female. That means whether a child will be a male or whether a child will be a female, this is determined by these chromosomes only. Okay. So during the gamete formation, okay, from these, can you tell which one is the dominant one? Which one will be the dominant one? X1, X chromosome or the Y chromosome?
X chromosome.
Yeah, X chromosome is the dominant one and Y chromosome, Is the X chromosome dominant or Y? Y chromosome is the dominant here, not the X one. See? So the gametes which will be formed, in the case of male, there will be two types of gametes. There will be an X type of gamete and there will be a Y type of gamete. Okay. So these are the two types of gametes which will be formed in the male, and in the female also there will, in the female, how many types of gametes will be there? Sorry, how many types of gametes will be there in female?
Sir, two, sir, X and X.
Two types or one type only?
No, sir, one type, sir.
Only one type of chromosome gametes will be there. There will be number of gametes will be the same, but there will be only one type of gamete which will be of type X. Okay. So now if the fertilization occur, if the zygote formation occur, so either the sperm containing the X chromosome will fertilize the X chromosome, or the sperm containing the Y chromosome will fertilize the will fertilize the ovum, right? So the ovum is just of one type, but the sperms are of two types, and their number will be same. If the sperms are produced, gametes are formed. So the gamete, what we mean by the gametes? In the case of male, we mean the sperm. In the case of female, gametes mean the ovum. Okay. So there will be just one type of ovum in the case of female, but in the case of male, there will be two types of sperm cells. If the X type of the sperm cell fertilizes the ovum, so you will have the formation of this XX, which will be a girl child. So you will have here the formation of a girl child. And if the Y chromosome fertilizes the X, so you will have the formation of XY, and this will represent a male child. Okay. So for either, since both of those sperms, they have equal opportunities of fertilization. So either of them can fertilize, and hence at the result we are obtaining a girl and a boy, or a male and a female. So we can say from here that both of them, they have equal chances of happening. Okay. So male and female, they have the equal chances, right? 50%, 50% chances are there.
Yes, sir.
Okay. And one more thing, a very important thing here, who is responsible? If the child is a girl or if a child is a boy, so who is responsible, the male or the female?
The
Male.
male.
The
It's the
male.
male who is responsible, not the female. Okay. It's the sperm which fertilizes the ovum, not the different types of ovums are there. There are different types of sperms. There are two types of sperms. Got it? So in this way, we are able to determine the sex of an individual. Okay. Now let us see about the blood groups. Shall we do blood group today or in the next class? Okay, let's see. What are the different blood groups that are there?
A, B, O, and
We have
AB.
the blood group A, we have blood group B, we have blood group AB, and we have a blood group O. Okay. And they are classified as these types depending on the type of antigen and antibody which they contain. Antigen and antibody. So which antigen is contained in the blood group A? Which antigen is contained in the blood group A?
What is meant by antigen?
See, antigen and antibodies, these are two different types of proteins. Okay. So these are two different types of proteins. Antigen is a protein, antibody is also a type of protein. Antibodies are the proteins which help in fighting with the diseases. Okay. So in the similar way, there are antigens also. Apart from them, you will also find a rhesus protein. A rhesus protein is also there in the blood, which is indicated by its nature as Rh positive or Rh negative. So you have heard that someone has a positive blood group and someone has a negative blood, right? So the positive blood group is indicated if it has a rhesus protein. If the blood has the rhesus protein, so we say that the blood is Rh positive. And if it lacks the rhesus protein, so we call that blood group as rhesus negative blood. Okay. Positive and negative do not mean the charges of the blood. They are not the charges. Okay, they are on the basis of the rhesus protein, which was first extracted from a monkey called rhesus in South Africa. So in the African countries, where their rhesus protein was determined, and hence it was also found in some of the human beings. So from then we started with blood group as positive and negative also. Okay. So by everyone, we'll continue the discussion about the blood group in the next class. Right. Okay, bye everyone. Good night.
Yes. Thank you so much.
Thank you, sir.
Yes, sir. Bye, sir.
Thank you so much.